today in the other lecture. Please feel free to interrupt and make all the questions you want. Make Jordi run. So that is up to you guys. He needs to do his exercise. He lost his morning exercise. So okay, so let's do this. So yesterday, that's the last thing I told you about, Huxley-Huxley model. And what we were saying is that, well, these guys were great, 52, and they developed, like, this was amazing work, okay? So they are heroes, okay, first of all. But nowadays, the problem is, well, okay, so now you have to decide which model you are going to use to model a neuron. And so so the question is, are you going to use this model or other type of model? That is the question, because when you go and say, well, hello world, I want to make a model, you have several choices, okay? So you have so many different models. So today I wanted to start the class just telling you about what you have to look at what model, when you are choosing a model, like when you go shopping, what you are going to choose, okay? So these guys are heroes, but the thing is that it's a 4-dimensional system, so it's difficult to study the dynamics. Also, because you have so many parameters, right? So these guys measured all these parameters, but again, this was 52, people didn't have internet, so they had plenty of time, and they were like recording like crazy. Okay, so this took them several years. I don't know if any guys of you know how many years they took to measure all that. They developed the technique, but it was like 5 years of recording, 10 years, okay? Because they measure all these values they measure, okay? So the thing with the dynamical system is that it really depends on the values of your parameters. Yeah, they measure the parameter values. And so the thing is that if you want to, you are using another neuron, so not from the squid, but from a mice or a bird or whatever, you have a lot of parameters to be determined. So you have to measure all these parameters. So having a full model with many parameters is not always the best thing to have. I mean, it's a thing. So this is what you have to know. If you want to use this model, you will have a lot of parameters, and also you have a neuron, and you have to know what channels you have. So you have a lot of modifications from this original. So you have a lot of modifications now saying, well, if you have a sodium channel or a different kind of potassium channel or whatever, so each neuron can have different channels and they will behave different, okay? Okay, so another approach is that you can go to lower dimensional systems. So if you want to reproduce a spike, you can use systems that are 2-dimensional systems. So for example, what the first step to do this is say, well, let's get a Huxley model. But what we are going to do is, so remember the equations. The equations in the HH model is you have this. So you have a fast current that you have M and H, a leaky current doesn't matter, and a slower current here. So this first approach is, well, this is so fast, let's do it instantaneous. So this has no dynamics. It's represented with a function of B. And then the only thing that I'm going to be modeling is that potassium current, and with just N, no N to the 4th, okay? So with this, I wanted to, okay, this, da-da-da. So how do I study this? So this is going to be a tour de force nonlinear dynamics. If you want to learn more about nonlinear dynamics, we can do this in the hands-on space. But for the people, so how do we analyze this? If you have a null clean is just a curve where x dot is equal 0. So for example, this system that has a two-dimensional system, you have two null cleans, okay? So the V null clean is going to be like all this equation equals 0, that is here. And you, so these are all functions depending on V, but now you will see why you want to get like n equals to a given function, okay? You do the same for the other one, for the n null clean. This is simpler, and you have only this equation. So two-dimensional system, two null cleans. And what do we do with that? We have this, and then here is where we know we have a spike. So this is, again, as yesterday, a first phase portrait. You have B and N that are the variables of your system, and this is the null cleans. Why do you have this shape, this N shape? Because this is the shape of this function. It doesn't matter. And this is the N null clean, okay? And here with arrows, I'm showing the flow. So the only thing in the N null clean that you have, so who understands this graph? So, yes, you better. You too, you better. Great. So who doesn't understand this graph? Okay. If you want, I can explain you like really in detail in the afternoon, or you can do that. So don't feel forced, okay? I don't need to explain you. But the only thing you need to know is, for example, in the N null clean, here is N. The condition of the null clean is that you have N dot equals 0. So the derivative is 0, so the arrow has to be horizontal, okay? Because this is the N axis. Do you understand that? Is that clear enough? Okay. And yes, right? Is that okay? No, I don't know. I don't know if I'm losing you, if I'm boring you. We are fine. Okay. 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 If you look in the V null clean is also, but with V dot equals 0. So here the arrow has to be like in this direction, so completely vertical. So how do we read this? So here is a stable point. So this system has one only fixed point. So remember the spike. I really like this blackboard. Do you remember from yesterday, you have the spike, and you have so this is one stable fixed point. So this is T, this is V, and this is the stable fixed point. Okay? So this is what we are seeing there. So you have just one fixed point, and then, for example, if you start with this initial condition, so for example, this value of P and this value of N, you will go all the way around, like following the arrows. Okay? Imagine you are like in a flowing. So it's called flow because it's like water, okay? So you get your little boat and go like, wah, until the next point, okay? So this is the spike upstroke, downstroke, the absolute refractory period, the relative refractory period, and then you get, okay? So this is our spike. So what I'm telling you with this is that just with two dimensions, you can get a spike, okay? So you don't need 4 dimensions if you want a spike, okay? So that is the concept. And here is the detail of the Fitz-Nagumo model, because my class should be before Christina's today. She just mentioned the Fitz-Nagumo, and now I will show you. So she presented these equations. I think Christina added some noise, something like that. But what is the core of the Fitz-Nagumo? The core, this doesn't matter. The core is this, that if you look at, again, at the nucleins now have this cubic form. And so A will shape if this cubic form, so okay, if you have a cubic form, this means that you have something like well, doesn't matter. But what I just wanted to show you is that you can have a cubic thing that is like this, or you can have, for example, something that is like this. Okay? And this shape is where you have a positive value near the origin. Okay? So whenever you have something like this N shape, you will have a spike. You have a dynamical system that is able to have a spike. Why? Because this N shape, when you cross it with the other nuclein, you will have the same thing as before, spike. You start here, go all the way to the spike. Okay? Any questions? Super easy. So this one is even with less parameters than before. So this is the core of Fitz-Nagumo. So you have just little amount of parameters, A, B, C. That's it. Okay, 3 parameters. And this is much easier to fit. You just need, so you go, you measure a spike, and then you see, well, how do I have to put A, B, and C to replicate the shape of the spike? Done. What I was telling you about yesterday, you don't need to know all the details of the currents. You just need to reproduce the dynamics, and that's it. Okay? So this is the strength of nonlinear dynamics. You don't need the details, you just need to know the dynamics. And you can go like really simple. 3 parameters instead of, I don't know how many parameters in HH. So the other thing is that this model also allows you to have a B stable. So if this nuclein is a relationship between B and C, okay? So the slope of this can change depending if you move the parameters. So you can also make this slope such that you have 3 fixed points. You have this kind of thing, you have B stabilities, that means that you have 2 fixed points. Okay, so depending on what your neuron does, you can also get this. Moreover, so this is an example where if you even care about the shape of the spike, but it can also be the case that you don't even care about the spike. If you just care if it's spiking or not, that is what I'm going to show you now with my data, maybe you don't even need. So if you just want to know if this has crossed the threshold and you have, yes, a spike or not, you can go and do even simpler models, okay? So integrate and fire or whatever, other models that probably some other guys will tell you about. So great. So this was just to finish with this nonlinear dynamics and neurons, okay? So this is my stop in the classes of nonlinear dynamics and neurons. I wanted to show you some data. So now that we have models for spikes, we want to record from real spikes, okay? And to see what model do I want, okay? So the question is, or is it the other way around? So probably it's the other way around. first we make recordings of spikes, and then we propose dynamical models to make sense of our recordings. So this is our approach, or my approach. I speak like Maradona, right? So our approach is my approach. Okay, so this is my approach. So I first record, and I use nonlinear dynamics to make sense of what I'm recording. Why? Because I'm just measuring few spikes, and as I'm measuring a whole brain that have thousands, millions of spikes, if I just measuring one by one, I cannot even make sense of what I'm. If I don't have models, it's really hard to me to make sense of what I'm recording. So that's, okay, so let's measure. So how do you measure spikes in South America, where sometimes you have money? So in Argentina, it's like this. Sometimes we are like champions of the world, like now, but then you cannot even classify, okay? It's like the football, like a football championship. So it's like this. It depends on what year you have. Sometimes you cannot even classify, and then you're champions of the world. So in Argentina, that means that sometimes you have money, like a couple of years ago, and sometimes you don't, like in the next years, probably. The cool thing about nowadays, is that before, like, I don't know, 20 years ago, you need to be like really rich to make neural measurements, like hundreds of thousands of dollars, okay, the equipment to record many channels. Nowadays, your budget needs to be like under $10,000. That it's not so much, so it's not a big startup fund. Why? Because you have, so this is what I have, so I will tell you what I have, but now you have so many companies that you can choose, but these are all the same idea. Actually, in TAN, what does is the chips. Then you have like open EFIs, for example, that is like the cheaper version, so they get the chips and make cheaper things. Or if you know electronics, you can buy the chips and all the other stuff, build it yourself, okay? So this is like $6,000, pretty cool, and you can record up to 1,000 channels, okay? So 1,000 euros. So I wanted to show you this because it's not only the input for the neurons, you also have other stuff. So analog input, analog output, digital input, etc. So you have so many things. Audio out is because you want to hear your neurons. So who has hear neurons here? Who hear neurons? Fiamma, you don't need to ask. I know you have. You don't need to answer. So, okay, so many people have some hearing neurons. Great. This is going to be so good. Okay. So, okay. Okay. Sorry about that. And then you have your electrode. For example, Neuronexus, but again, you have so many other companies. I'm just showing you what we have. So this Neuronexus Pros, it's also kind of modern. I don't know, also like 10, 15 years ago, something like that, we started to develop. So these are micro machine silicon probes in the sense that this is an electrode and you can design. So these are different types of electrodes. So depending of your animal, of what you want to record, you can order different electrodes. And for example, you can also order these. So chunks, where in each chunk you have different electrodes. And so this you can design. You have a lot of, so you have a catalog and you can choose. Actually, what you do is you ask your fellow biologist friend what electrodes you are using, and he's saying, yes, I tried this and this works and this not, and then you buy those. But if you want something special, you can also order those. These are the real things that you can implant. So this is the electrode. So the thing is that, and the value is around $1,000. So again, so depending on your budget, this can be okay. But what I was telling you before, you just need $10,000 and you can, let's say, $20,000 and you can do recordings for pretty much many years. So if you have that kind of money, you just plug, so this is the amplifier, sorry. So the thing with the recording neurons is that these are very small potentials, okay? So you need to amplify your signal, and the thing is that you work in a Faraday cage. You know what a Faraday cage is? Okay, so it's this guy Faraday that was really crazy. He invented this cage. Say, well, if I put metal, I can have a lightning and I won't be killed by this lightning. So he did that. So inside of a metal cage, you are kind of isolated from noise from outside, so nothing can penetrate you. You work with a Faraday cage, but if you have an amplifier that it's really close to your electrode, that's much better, because when you go out of the amplifier, then you can put a long wire and it doesn't matter, okay? So you plug your electrode with the amplifier and you start recording. So what do you do? Yes. Sorry, louder. What? The signal? In Portuguese, you can ask in Portuguese.
A digitalização do sinal.
Oh yeah, but yes, the thing is that this is all digital. That's the great thing. So this is already digital.
Yeah, but at which stage? Like together with the amplification, or?
Yeah. So after the amplification, it's already digitized.
It's already.
Yeah. 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 That is super cool. Also. Yes. Okay. Any other questions? Okay. So that's it. So come on. So if you don't have that kind of money, or again, if you are in Argentina and you know, so this was my situation. I returned from the United States and we were rich back then. We were the champions of the world in Argentina, and I bought a lot of electrodes. And we still have them, right, Fiamma? We still have them in the lab. She can tell you all about it. But I said, probably this is not your problem, but I don't know. This is my experience. I don't know if it's really helpful for you guys. In Brazil, it's everything more steady, right? You know if you're going to have money or not. In Argentina, you never know. Okay. But I knew it's going to come a day where we won't have any money. So again, other people in Argentina. So you have a cheaper, so this can last forever. So you can manufacture your electrodes. So one guy's Busaki, it's some guy from Europe, East Europe, that went to United States. So he developed a lot of electrodes and things to measure. And what they discovered is that if you put 4 electrodes together, that it's good enough to separate neurons. I will tell you a little bit more about it later. So you can make your own tetrodes. You have a tungsten wire, very thin, so 12 microns, and you can make your own electrodes. And so what we did was develop a machine to record from the brain. So this is a screw, and here are the tetrodes. So we are using 4 tetrodes, so 16 channels. And with this screw, you can go up and down with your electrodes. And here is the connector where you plug your amplifier, okay? And all of this you mount in the bird. We work with birds, right? I told you before. But you can mount this with rats. So depending on the animal, you have to design your electrodes. So birds cannot carry so much as a rat or a mouse, but they are less strong than a mouse of a rat. So a mouse, it's like going to take out. The bird cannot do that. So it's 14 grams our birds, or 20 at the least. Okay. So we designed this. So this is, Fiamma works with this. So she makes the electrodes. This is our device. And what are the advantages of this? They are cheap, but they are ultra lightweight. And you can build your electrodes as you want. So we are just recording 16 channels because the bird cannot carry so much weight, but you can do as many channels as you want. You can also, for example, this is the real thing. And so here are the tetrodes you can see. So we have 4 tetrodes, and this is a reference. But if you want to change your geometry or well, I'm going to use 2 tetrodes here and 2 tetrodes there, measuring different regions of the brain, you can make it, you are building it yourself. So that's the only advantage of building it yourself. Like when you build a house, you build it at your own desire. No. These are the electrodes. So this is just for you to see. So we have the tetrodes, so 4 tetrodes, and we cut them. And what you're seeing is this cut. So it's this 1 electrode, 1 electrode, 1 electrode. So how you look, yeah. Okay. So. Yes, so what you're seeing there is a cut like this. And so, for example, maybe here you can see it. So here are the 4 tetrodes. And with the screw, what you do is that you go deeper in the brain. So you implant it, and then so you put all dentin cement here, and then you go with the screw every day and move the screw, and you go like fishing neurons. Okay? You can ask Fiamma about it. I can answer that too, but let's make her, okay? This is the device. Come on, the other button. So those are the recordings, okay? These are the recordings we obtain, the raw data. This was done with a former student, Santiago Boari. So you can low-pass filter the signal, and you get what we call local field potential, so the slow part of the signal, okay? Or you can make a high-pass filter, and you get the spikes, okay? This is an example to making this super obvious of what is going on. You can see the spike. With this signal, you don't even need to do anything, okay? It's so powerful. In general, you don't have the waves so clear, and the spikes are more smaller, right? Like this one. But this is what you can do. So with your neural signal, you can have the local field potential or the spikes, okay? So I will talk first about the spikes, and then I will compare those with the local field potentials. Okay, yeah. Okay, so these are the spikes. So what do we do? So here is what we call multi-unit recordings. So this is one electrode is picking this signal, and this works like this. Like you have your neuron right here with the soma, and you have your electrode here, okay? And for example, and you have another neuron here, okay? And you're doing extracellular recordings. Okay? So what are these spikes? So if these are extracellular recordings, so this is what I have. I have the electrode that is outside the neuron. So what are those spikes? Sorry? Yes, they are talking to each other, but the question is why you see this, why I'm saying that this is a spike. What is the recording? That's what I'm saying. But like if you are inside the neuron, you measure different ion currents. But if I am outside, why do I see spikes? You understand the question? So it's a very basic question. Exactly, because we have chemical connections. That means that you have ions go when these guys spike. What they do, so here is some when this. No, we have this one, okay, and this is receiving connections from other guys. No, actually, this is what you are seeing. Okay, so a neuron doesn't spike, but gets fired by other neurons. So here, you have chemical connections, so you will have ions in the bath. And what you are measuring here is these ions, these currents. This is why the currents are really small. This is why you need an amplifier, because these are few ions, okay? Okay, so now we know what we are measuring. But what I wanted to focus here is that you can see you have different amplitudes. So you have bigger spikes and small spikes, okay? This relates to the distance from the electrode. If the neuron is really close to the electrode, you will have a bigger spike. Not because the spike is different, but because you are measuring like closer. Like you put a microphone, okay? So if I put a microphone near these guys, I will hear what they are talking. But if anyone is talking there, I won't hear, okay? It's like a microphone and people like whispering, okay? So that is recording. That is neural recording. Okay, so any questions? Do you want me to ask? Do you want to ask me a question? Yes, come on. Yeah, you can do it. Yes.
We didn't understand why we measure only the ions and not the dipole of the axons.
You have to speak louder.
Okay. We didn't understand why we measure only the ions and not the dipole, the difference of potential. Why we not measure this and only the ions?
Well, that depends. Yes, you can think. You can also think it like you have a capacitor. So that depends. So, but the basics. What I wanted to go to the core, and say you are measuring. So this is voltage changes. And you have a voltage change because you have an electrode here and a ground somewhere else, okay? So, yeah, so you can think you have ions, or you can think the neuron as a dipole. It doesn't matter. But it's all about, so what I wanted for you to think is that you are measuring this. When we say potassium currents or sodium channels, those are ions with charge. And this is what you are measuring, okay? And the ions cannot be, or for example, if you have ions that are chloride, those are negative. So you have all these different ions. So I wanted you to think in ions, but then you can think in dipoles. That doesn't matter. Yes. Another question? Yeah, we are going to the core, so I'm just showing you data. So it's cool. For me, it's better if we discuss this and you kind of understand.
Okay. I think Georgie yesterday said that there are some like fake spikes that the neuron doesn't intend to release the ions to connect to another neuron. Can you see what the fake spikes is in your data?
Do you want to clarify that for me?
No, what I said
the fake spikes?
No. Well, what I said yesterday is that you have to do triangulation techniques to detect which is the neuron that is firing. So that was related to one comment that do you have access to the location of the neurons in space, or you do not?
Well, actually, triangulation is what we are doing with the tetraodes. Actually, because of that, is that we have, so this guy kind of show that maybe with 3 is enough, but 3 it's very difficult to build. So we just put 4, and it's very easier to fill. So, but if you have 4 tetrodes, I really like this blackboard. This is why I am. So you have the 4, let's do it bigger. Okay, so you have, so this point, so actually what you have here is these are insulated, and you have the tip, so just okay? So these are insulated and you're recording here, okay? So this is why you use, so this triangulation actually is what we do. Because if you have your neuron here, or your neuron here, this neuron is much closer to this electrode than this one. And if you're recording from 4 channels altogether, so if you have just 1 channel, you don't have any relations to where they are. So you can separate individual spikes, because that's the thing. So you have multi-unit recording, and it's all a mess. And then what you want to say, you have 2 options. Either you get with that mess and say, well, I'm going just to get everything multi-unit, won't separate which neuron is which, or you want to isolate neurons. If you want to isolate neurons because you want to know this particular neuron what it's doing, you have to triangulate, exactly what Jordi is doing. The thing is that we are not in a dish. We are in the whole brain. Why we want to triangulate? Because we have different kind of neurons. We can have excitatory neurons, inhibitory neurons, different kind of inhibitory neurons, different kind of excitatory neurons, and we want to kind of tell apart what kind of neuron is doing what, okay? Okay, any other questions? Okay, yeah? Great. Yes?
Now that you talk about excitatory and inhibitory, the Figus-Nagumo model, which type of neuron represents?
Well, the thing is that a spike is a spike. You don't know. So it could be excitatory or inhibitory depending on the ions that it will expel when it has the spike. So a spike is not, to me,
Spike
excitatory.
is not a priori excitatory or inhibitory.
Or inhibitory. It's the ions that released it. Do you guys agree?
No. I
Is
agree.
that okay?
I agree with you. No. It's
Great.
you cannot know.
You cannot know.
You see activity.
Yes.
You have to indirectly try to assess if that was excitatory or inhibitory.
Yes.
That's why I ask if you have any way to put a camera. No, it's not possible to put a camera.
Yeah,
and
no,
see.
but the cool thing about birds or the region that we are recording is that inhibitory neurons have a tonic firing, so a particular way of firing, and excitatory neurons are phasic. So we can tell apart with the firing rate. So we know if it's an excitatory or inhibitory because of that.
Now, let me make a comment, or a question. Can you stimulate with the electrodes so you can interrogate the circuit locally?
Yes, you can. We don't do it, but you can do it. Actually, what you can do is put, I would say you can put in the same region. Yeah, so you can do it.
Yeah,
we don't do it, but I think, yeah.
Yeah, I'm asking the question because in the context of what I explained yesterday, if you interrogate the system, you know the behavior of the neurons in response of your stimulation, so you can know if it is excitatory or inhibitory, or with which other neurons is connected to, and so on. Yeah, so you get a little bit of idea of which is the circuit that you are investigating, right?
Yes, yes. We don't do it, but some other people in our community, what they do is, so this excitatory, they connect to other neural nuclei. So, but in this region, you can connect to different kind of nuclei that do different things. So if you want to know if this excitatory neuron is connecting to this region or to this region, you put an electrode in another place, and then you make an excitatory pulse, and if you can pick it up in your neuron, because you have like retrograde information going, you can know if the neuron is projecting to one place or to another. So yeah, you can do that. Yeah. Okay, cool. Any other questions? Okay. Okay, so this is the thing. So if you have just one channel, that is what I'm showing you here, the only thing that you can expect is you have a really good electrode like this one, and you have like very close neurons and faraway neurons. And you can separate those depending just on the amplitude. So for example, if you put a threshold here, each dot will be on a spike of these bigger neurons. And then you don't count these smaller ones. So this is what we call spike sorting. So crossing, the simplest method is threshold crossing. So if you have a good signal, you can put a threshold and you can separate these big guys from these small ones, okay? And then what do you do with that? Well, then you build what we call raster plots. So each of these dots goes in this line, okay? Here, each dot is one spike. And these are trial numbers. So as you can see, here we are presenting some stimulus. So the neuron is firing like in regular fashion. But here something different happens. And what is happening is that we are presenting an auditory stimulus. But you can see that the neuron is responding to that. So it's responding really strongly to that stimulus because, and you see it in the group data. Okay, you need statistics when you are measuring. It doesn't count if you are just measuring one. Whenever you are measuring in nature, you need statistics. Okay? Nice. But then if you look in it, again, another example, but if you zoom in, you also can see that you have different shapes. It can only be because if you don't have such a clear data, what it generally happens is that all the spikes are more or less the same amplitude. So because your neurons here are kind of far away. So you have another way of doing spike sorting. That it's, you can do PCA analysis or whatever. Now there are a lot of tools using artificial intelligence, but the idea is that you can separate different, you can see these are the shapes of the spikes, that they are very different, and what you have is, we call it also cluster cutting, because you have a cluster of your neurons, and this is like highly dimensional, so this is a two-dimensional plot, but then you change the axis and you have like little clouds and you kind of separate your spikes like this. Okay? So from one recording with a tetrode, you can have individual spikes. Okay? So it's very complicated and it's very hard. You can ask Fiamma. It's really hard to get data. Okay? And it's a lot of work to get the data from the experiment, and then you have to analyze it. Okay? Okay, I wanted to show you this. So this is the real thing. So what you are, so this is a video of a former student, Cecilia. She was so happy she was having her recording, so she just took the phone and recorded the monitor, okay? So this is the bird singing, and these are the 16 channels. So each of one of these is a tetrode. And here you see that, for example, this tetrode is picking up this big neuron, and these are not, okay? What I was showing you before. And these are another examples of previous recordings, but what you will see is here. And hearing the neurons, so each spike is a click. This is why you can hear spikes. And these are voltage changes. So if you connect this to a speaker, you will, whenever you see a spike, you will hear a click. And this is so cool, because we are visual animals, the humans, we are very visual, but when you start hearing, you can hear much more subtle differences than what you can see. And so when you're doing electrophysiology, you hear spikes, you don't look spikes. Okay, so what you can see there is that it was singing at the beginning. You can see a lot of activity in these tetros, and this was, I will play it again now, but this was synchronized with what the bird was saying. I don't know if you could hear it, that the bird was doing like, pa, 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 and the neurons were doing like, go, 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 okay? And then when the bird stopped singing, these neurons kind of say, well, okay, I've done my work until this guy wants to sing again, I won't do it anymore. Okay, you can see that like, pa, pa, pa. Those are movement artifacts that are the worst enemy that when the bird like jumps, because the bird is like freely moving, right, in the cage. When they jump, it's like, okay, so this is what we do. We implant the microdrive that has to be very light. We train the animal to carry those, and then we record neural recordings when they are singing, okay? Or we play back a recording of their own song, and those neurons has this property of also firing when they hear or when they are singing. These are like mirror neurons in macaques. They fire in the same way when they are hearing and when they are singing, okay? The cool thing about these birds is that they are temporary lock. So they fire in the same place, not in the same fashion, in the same place when they are hearing or when they are singing. And if you think about it, they are doing very different things, because when they are singing, these neurons should be like premotor. So this should be like before the motor action. But when they are hearing, the stimulus came before. So in a way, they are like kind of synchronized so that they fire in the same place. So these are very cool neurons. We love them. Okay, what else? Okay, I just wanted to show you a couple of things, but this is our kind of what you can do with data. Doesn't matter. So we can stop here and you make some other questions, or I can show you more data. It's up to you. I don't care. Yes.
Recorded the neurons from different species of birds?
Yes, yes. So that's a cool question. So the cool thing about birds is that we have 4,000 species to pick from birds that learn to sing. So tomorrow I will tell you more about it. But different species do different things. And in our lab, we work with 2 species that are very different, zebra finches and canaries. The song is very different. The behavior is very different. In canaries, you have adult neurogenesis. So each spring, they learn new songs, and this is related to neurogenesis. So in autumn, neurons die. With testosterone, in spring, neurons born again, reconnect, and you have new behavior and new neurons to record from. Yeah, they are super cool. So those are canaries. Not many people work on them because they just breed once in a year, and they are more hard to sing, right? So to make them sing, it's like Fiamma has to do like all this thing that, come on, guys, sing. The other species, zebra finches, they breed during the whole year. You have an adult bird in 90 days. You have a very sophisticated song, very different. And if you, in this species, only the male sings. There are other species where both male and female sing, but in this species, if the male is singing, and if you present a female, the male will start singing at her, like, yeah, 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 yeah. Like, look at my wonderful song, come with me. So yeah, and the canaries don't do that. They don't care. No, the recordings are very similar. That's the cool thing. So neurons, what we know so far is that they have the same neural nuclei, the same type of neurons. There is a question of why they sing so different, why do they have different behaviors? Because what we've studied so far is that they are all the same neurons.
Yeah, because I remembered a lecture that I saw on SFN in 2016, and the presidential lecture was about bird singing, and they exchanged species. So they put like a baby bird with another species, and this baby bird got like an accent.
Yeah, that was Sarah. That was Sarah.
Yes,
Sarah Woolley.
I just confirmed here.
Yes, actually, yes. So those are very cool experiments. So she does cross-fostering. So these birds learn from a tutor, and so there is also they think how much they learn from a tutor. So they have something that it's innate. So it's not that every species can learn anything. It's not that. That depends on the bird. But they can copy. So if you put two closely related species, they can learn from different tutors. So those are cool experiments. So if you do cross-fostering, what are the changes? And the cool thing that she finds is that they have a constraint, is that they can copy some things from the tutor, but they are constrained by the biology. But we don't, so it's not that they can copy all the things. We don't know what constraints are. We don't know there is some innate biological constraints, but we don't know what it is. Are the ion channels? Is the network structure? Is something else? Is the glia? We don't know. There are differences, but what we have, this is why I told you, what we measure so far, we find at the level of spikes, they are very similar. That's so much to know already. So come to work in birds. Yes, we need more people.
I would like to know that they're the same site, anatomical sites, they're electrodes.
Yes, we have atlases, we have like maps and see, well, from this point, go 400 microns left, 200 microns deep, and then you will have it, yeah.
But where are they exactly? Like, is it more frontal or occipital?
Oh, okay, these are like cortical regions. So there's all this discussion because birds are dinosaurs, okay? So we want to say they are very similar to mammals, but they are dinosaurs. But they have structures that are very similar, so there is a really strong fight if they have cortex or no. So for sure they don't have neocortex. That is established. But we, the bird people, say they have something very similar to cortex and also to auditory cortex. So the structures are different, but the connections are very similar. And these are cortical regions. And the difference from mammals is that they are not in layers. They are like nuclei. They are like balloons, something like that. Yeah, I don't know. I can show you some pictures later if you want, so you can see the balloons there. But I don't know if that was your question.
Yeah, no, I just wanted they are located in auditory cortex, probably, so.
Okay, no, so these neurons are, they receive auditory input from the auditory cortex. So the highly processed auditory cortex sends information to this region. And this region also form parts of a motor network. So this is why they can do this mirror thing, because they receive auditory input, but they are essential for singing. If you lesion this nuclei, the bird won't sing. Tries to, but it's not good.
Thank you.
Another question there? I don't want to finish my lecture. I have another lecture tomorrow, so yeah. Oh yes, the brain. I don't like that brain because it's so different. Tomorrow I will show you a picture. So we don't have, the brain in birds doesn't have all these things. So it's like more, it's liso. I don't know, it's smooth. Yes.
Yeah, something that I wanted to discuss with you is that when we hear words, so this is basically our auditory cortex, okay? When we hear sound goes here, and here this is processed. You see this green dot over there. You see this green dot here. This is where you understand the words. Okay, so the information is sent from the auditory cortex here. So this is called Broca's area. There you make sense of the words, and then you send it to the prefrontal cortex, where you put it in context of what you know. Okay, so it's very complicated. But then when you speak from the prefrontal cortex, you send information to the same area, so you build the corresponding words from your knowledge, and then you send it to the motor cortex, where you synchronize everything to make that word. So it's, in mammals, it's extremely modular and complicated. So what happens in birds?
Well, one thing that happens in, so what he's describing, this is lateralized. So this happens in one region, in one hemisphere, but not in the other one, okay? In birds, we don't know. So the first guy that started studying this, it's called Fernando Nottebohm. He's Argentinian, working in New York. We are not so good friends with this Argentinian guy. He's kind of, well, I don't comment on that because we are being recorded. We love Fernando Nottebohm. He should get the Nobel Prize because of his discoveries in neurogenesis. So he started studying with canaries because of this adult neurogenesis that I told you before. He started working with a brand, I don't know, una raza de canarios that it's called Waterlager. So Waterlager. So it's una raza de perros. I don't know the name, raza. So it's like when you have a Doberman or a Rottweiler breed. Okay. So this particular canary, it was lateralized. So he was just seeing some activity in one part of the brain. So he was saying, well, it's the same as humans, they are lateralized. But then, if you look at another type of canary, so the canaries that we work with, so it's not this specific, but it's another one, so it's common canary. It's not lateralized. So in both hemispheres, happens the same. They are interconnected, and they are not lateralized. And in other species, zebra finches, in all the other species that we've been recording, they are not lateralized. So this lateralization, it could be a specific thing of I don't know, it's very specific. But the thing of semantics, if you say, so we are just focusing in birds with syntactics, if you want. So we are studying birds because of the vocal learning, because they can learn from the tutor, so vocal learning is very weird in different species. But we are just saying, well, you can do the motor command and you learn to vary your vocalizations from a tutor. But we are not studying the message communication. So we study song because these are very, so they have to coordinate a lot of muscles, respiratory muscles, like vocal apparatus muscles and all that. So you have to be like very dexterous to perform a song. But most of the signals are communicated through calls. So the message is, it's more like simple vocalizations and say, for example, and they study that. So they had different calls for example, the experimentalist is coming inside the room, they have a call for that. And so they are kind of, as monkeys, these experiments that when they have some calls for there is an attack from the sky or there is a snake, so they have different calls that is the same. So these are not necessarily learned. So all the semantics is not well studied. We have also some species, starlings, they are called European starlings. That species, both the male and the female sings, and those are like very complicated vocalizations. So in that species, you want to study semantics. The thing is that it's so hard to study because they can have like 60 different vocalizations. They can remember these vocalizations for years. So it's not that all the species have the same level of complexity. So if you want to study about semantics or something more sophisticated, you have to go to these other species, but they are much harder to understand. As crows, for example, these crows that they can make tools, they are like super intelligent guys, but you have to work with crows, right? So that is the thing. You have to choose a species depending on what you want. So we still don't, so the short answer to your question is we still don't know. Still, we have to do so much research to know. And, okay.
A philosophical question and
Yes.
a practical question.
Yes.
The philosophical one. Is there any significant difference between a neuron in a bird and a neuron in a brain?
I would say no.
Same neurons.
So, yeah, same neurons. So same channels, same, yeah. I couldn't think of a difference. So we are using the same models, if you want, like, yes, the same channels, the same, we don't see any difference, yeah, at that, yeah.
Okay. The practical question. The algorithm that you use to triangulate and to extract several spike sequences from just one spike sequence, is that commercially available? You did it, you sell it?
It's commercially available, and you can also put your hands on it. So it's commercially and it's open to the
it's
community.
open, so
it's
you don't
open.
have to pay.
Yeah, no. Okay, yeah. You can pay. Those are fancy things, but nowadays everyone is using with open source software.
Open source software.
Yeah.
So the two of you could use the same software to extract more or less?
Yeah. It's always with the tool. If I have my screwdriver, I will use my screwdriver and not his screwdriver. But it could be the same.
You tune your code, towards your...
But you don't develop from zero the code.
It can be difficult because sometimes, as she said, you work with a particular species. Sorry, you work with a particular species of animal, so then you tune your code, right, for that particular experiment. And sometimes if you want to share it, then you have to be careful because maybe some parts are specific to that problem, no?
Yeah, in the sense, for example, is that at the level unit, you can have whatever channels, but sometimes you have specific channels. So, for example, you have human spikes are like smaller. You get like smaller amplitude and more robust. So it depends on, for example, how the rate. So you can have like if you have a conglomerate of neurons. The software you need may be different if you have a more sparse. So that is the tuning that Jordi is saying. In principle, yes. So in principle, we can do it. But then this fine-tuning. So actually, our thing is that these are developed for the software that we use are developed, and the parameters are tuned for rats or mice, and we sometimes had to put some, adapt the parameters so that we can isolate our neurons. Yeah. Yes, we're really late.
So we should go on. Yeah. Thank you, Anna, again.
Okay.
Thank you.